Hello, good evening everyone.
Good evening, ma'am.
Okay. So this is our last session for this class. Next week you have your exams. So in this week you have studied mainly about the biomethanogen process and fermentation that is undergoing in IIT Kharagpur. Like they showed you the real system where they are performing this. So today's session is based on these topics also. So let's start. Okay, this is your first question. What they are asking that according to WHO guidelines, what is permissible limit of BOD in drinking water? So they are simply asking uh, for the BOD level in drinking water as per the WHO, because mostly these at the international level or globally, this WHO usually produce guidelines for different levels, level of different metals, different ions and other things. So in drinking water, what is the normal BOD level that is available or that can be permitted? Uh, that is around 6 mg per liter. So when you convert this or when you see this in terms of ppm, this means parts per million. This ppm means 1 by 10 raised to the power 6. Okay. So according to WHO, the level of BOD in drinking water is 6 ppm. Okay, when you convert this, it will become 10 raised to the power 6. So for this question, answer is A. Like only this is only a informative question. You only know like what is the limit allowed. Like here they are asking about the BOD, but they might ask about the some metals which are especially heavy metals, because they will contaminate the water. So the level of this need to be checked properly. Now coming to the second question. Okay. In this question they are asking which bacteria is generally used for activated sludge treatment process. So this is also a very simple question. Anyone want to answer this? Like which bacteria they will use in activated sludge treatment? Okay, first let's read the option. First one is Archaeobacteria, second is Juglo ramigera, third is Chlamydomonas renhardi, and fourth is cholera species.
Option B is Juglo
Ramigera, yeah,
Nigera.
this is correct. This bacteria is mainly used in activated sludge treatment, that is Juglo ramigera. So for this answer is B. All questions of today's assignment are simple, like little bit of information only that you know. So for this question, answer is B. Okay, this is your next question. The sludge volume index for wastewater treatment is the. So what is this sludge volume index represent? Okay, what are the options? First one is the mixing characteristic of the sludge. Second is the settling characteristic of the sludge. Third is the microbial load of the sludge, or fourth is all of the above. So this sludge volume index, what is this sludge volume index? This is the volume in ml occupied by 1 gram of suspension after 30 minutes settling. Means it helps in knowing the settling characteristic of the sludge. Means how much it will settle. So that we will calculate using this SVI. This will depend upon the composition of this sludge material and what are the components present, what are the other waste, or if in some of there is more precipitated compound, then it will have more SVI. So this SVI is mainly as a, we will use as a unit or as a term for this activated for measuring the settling characteristic, especially as it is written that by 1 gram means 1 gram of suspension after 30 minutes. Normally how much we desire SVI means how much SVI is accepted or considered good for the treatment, for this wastewater treatment. Normally if it will have 100 to 200 ml per gram, then it will be considered good. Otherwise, if it is more than this, then there is a problem in treatment, and if it is less, then there are chances that the molecules are remaining in the wastewater that is not treated properly. So that's why there is a particular range in which it should be there. SVI means, just a minute. They will consider time limit of 30 minutes for calculating this SVI. Okay, and they will see like how much ml or how much volume is settled in a per gram suspension. So if it is around 100 to 200 ml per gram, then it is considered good SVI level. Okay, so this sludge volume index is mainly used for or help in characterizing the settling of the sludge. So the answer is B. If anyone has any issue, then please ask me in between. Now coming to the next question. They are asking the purpose of coagulation and flocculation in activated sludge process is to, first option is remove microorganism, second is remove microorganism, organic matter and suspended fine particle, third is remove taste and odor problems, and fourth one is suspended fine particle. So anyone want to answer this?
B, ma'am. Remove microorganisms,
Organic
organic
matter and
matter.
suspended fine particle, yeah, correct. Both this process helps in removing all the three things. If even if there are fine particle, they are also removed. Microorganisms are simply removed out, and organic matter are also used using different components. Even sometimes they use different chemicals. Or microorganism role is always there. So both these processes helps in removing all these things. So for this question, answer is B. Now this is the next question. Okay. They are asking methanogenic bacteria have the presence of what? Okay, let's read the option. First one is isoprenoid rich membrane. Second is isoprenoid rich membrane lipid. Third is isoprenoid rich cell wall. And fourth one is membrane lipid rich in film. So they are asking about the structure or what is the like differentiating factor of the methanogenic bacteria from the other bacteria. So what happened in methanogenic bacteria? Like this is the example of methanogenic, this methanovibribacter. So this is the cell wall, sorry, cell membrane of this. In cell membrane, they have shown the lipid. So in lipid, you can see in this figure, in this part, that there is a branched isoprene chain is present in lipid, especially. This branch isoprene is a compound that is present in this lipid layer or a lipid part of this membrane, and they are linked here. They are linked here with the ether links, like the bonding is here is ether bond. But if it is not the methanogenic bacteria, what happen in the bacterial lipid, in a normal bacterial lipid which is not methanogenic, unbranched fatty acids are present. And they are linked through the ester bond. So this is the major difference in the structure of this methanogenic and non-methanogenic, you can say. In bacteria, the structure is isoprenoid chains are present in the lipid part and they are linked through the ether bond. But in the normal bacteria, there are unbranched fatty acids present and they are linked through the ester bond. So this isoprenoid component is present in membrane lipid. So for this question, answer is B. Suppose like in this question, if this second option is not given, they are not specifying that this is the membrane lipid, then you might take this cell membrane part. But if it is given clearly that it is present, this isoprenoid rich membrane lipid is there, then this is the correct answer. Okay. So now coming to next question. In this they are asking which of the following is not a class of methanogen. So previously we studied what is the distinct feature of these methanogens. Now they have given the types or class of this methanogen. So you have to tell which is not a class of methanogen. So first option they have given is CO2 reducing. Okay. Second is methylotropic. Third is acetic lactic. And fourth is acetobacter. So please answer this. Anyone?
Acetobacter is not a class of mutagen.
Yeah, correct. This acetobacter is not used for this methanogenic. So this acetobacter is not a class of methanogenic. So for this question, answer is D. Okay. Yeah, this is your next question. In this, they are asking, in case of activated sludge process, the relationship between hydraulic retention time and mean cell residence time is. Sorry. What happened? Like both these things are the time given or time required, you can say. Normally, from the name itself, you can clarify what they are asking. In first one, they are talking about hydraulic retention time specifically. Means something is retaining, and in the mean cell residence time, similar. This is the residence almost same type that how much time they are residing there. So here they are asking about the mean cell. So from the name itself, it is clear that hydraulic retention time is the time at which the wastewater will flow through this fermenter, for how much time it will flow. Okay, and the mean cell residence time, cell means what is the microorganism they are using, whatever microorganism they are using, so their time in the fermenter or how much time they are staying there. So normally we keep the microorganism for longer time as compared to the this flow in the, or this is wastewater. So what, from this it is clear that hydraulic retention time, it usually up to hours, it goes like 4 to 6 hours, but mean cell retention time, it will go up to like 3-4 days. So from this it is clear. What is the option they have given, that this HRT is equal to MCRT? No. This HRT is more than this mean cell residence time? No. This hydraulic time is less than mean cell retention time. So for this question, answer is C. Now this is our next question. They are asking what is the percent of methane in natural gas? Options are 40 to 70%, second is around 99%, third one is 75 to 99%, and D is none of the above. So how much methane is present in natural gas? Okay,
40
no.
to 70, ma'am.
Sorry?
A, option A, 40 to 70.
Okay. No. The methane is present almost more than 70%. Even somewhere you will see like they have given 70 to 80% or 70 to 90%. At some places they have given. But it is normally more than 70% or around 70 to 90%. So for this question, answer is C, that this methane is present around 75 to 99%. You can simply remember this, that it is more than 70%. So whichever is the suitable option, or in different literature you will get that some different values, but you will get one thing, that this is more than 70%.
So I got confused, ma'am, actually. I was thinking that the question was for biogas. So biogas, it is 40 to
Yeah,
70.
for, okay, yeah, here it is they are asking for natural gas. So for this question, answer is C. Here, this is your next question. They are asking which of the following metabolic pathway is applicable for biomethanation process. This biomethanation process, they are asking which pathway is used. First one is EMP pathway, second is Barker's hypothesis, third is TCA, and D is none of the above. So these two pathway, EMP and TCA, we have already studied in the previous assignments. So why this EMP pathway is used here, or for what this is EMP pathway for?
Citric acid.
EMP pathway
No, 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 no. EMP pathway for
for glucose,
alcohol, ethanol.
or similar like glycolysis.
Yeah,
This TCA is for citric acid.
yeah, yeah.
Okay, so for these two we already know. And this Barker hypothesis is mainly used For this methane process, you can say biomethane. So for this question, the metabolic pathway is Barker hypothesis. Answer is B for this question. As you have exam last, maybe next week you guys have exam on 29th. So try to revise all the lectures. At least go through your lecture material that is available in the NPTEL site. From that you will get a little bit idea of everything and you will remember the all the things that you have studied till now. Now this is your next question. Which of the following gas may be used for the Haber-Bosch process? First one is methane, second option is natural gas, third is carbon dioxide, and fourth is all of the above. So for this Haber-Bosch process, which gas it used? Okay, this Haber-Bosch process uses methane. Okay, so for this question, answer. Okay, this uses natural gas. Sorry, for this Haber-Bosch process, this natural gas is used. Yeah, for Haber-Bosch process, like for the synthesis, for the initial, they use this natural gas.
Actually, methane can also be used. This Haber-Bosch process is for synthesis of ammonia.
Yeah,
NH4.
correct. Like, but
So we require hydrogen. Methane can give hydrogen. Natural gas also can give hydrogen.
yeah, it can also be used, but mostly people use this natural gas or they prefer natural gas because in this
Of course,
the
natural gas is
percent
also
is
a kind of
more, so
methane.
that's why. Yeah, correct. That both of the thing can be used, but preferably natural gas is there. Okay. So this is from your last lecture part, that which of the following microorganism was used for hydrogen production under dark fermentation at IIT Kharagpur. So the which bacteria they are using, they are, from the name itself you can see that this Enterobacter cloacae, IITBT08 and Klebsiella pneumonia, that another strain they have used. Normally this, for this question answer is B, but this name because they have named these microorganisms or they have discovered to use this. So these bacteria named, they have named them with their signature. Normally you will get these type of signature that will help in identifying the place or particular person name that will help, that has identified that microorganism. So they usually named in this way. So for this question, answer is B, that they have used this Enterobacter cloacae and Klebsiella pneumonia for this hydrogen production. So answer is B for this question. Now coming to the next question, this is what they are asking, that according to stoichiometry, how many moles of hydrogen are produced from 1 mole of glucose? So what are the options? Okay, let's first check in this that this is normally the metabolic pathway that they have glucose or any sugar source. So when it will be converted to pyruvate, this is glycolysis process, then it will further convert it to acetyl coenzyme A, and if it will form, here they are asking from acetate, yeah. So if this acetyl coenzyme A is converted to acetate, then it will form the 4 hydrogen, like 4 molecules of hydrogen. And if it get further converted to acetoacetyl and finally to butyryl, so when it converted to butyrate, it will form 2 hydrogen molecule, because they are only asking about these two, that either from acetate and second one is butyrate. So we are considering here these two part only, that when this glucose is converted to pyruvate and acetyl coenzyme A, this 2 acetate is formed and this will form the 4 hydrogen molecule, and if it is from butyrate, it will form only 2 hydrogen. Okay, so for this question answer is, let's read it. In first option they are saying that 2 moles through acetate and 4 moles through butyrate pathway. Okay, 2 moles from acetate, no. From acetate we are getting 4 moles. Second option is 4 moles through acetate, and second is 2 moles through butyrate pathway respectively. 2 moles are produced in both the pathway, no, and none of the above. So for this question answer is B, that 4 molecules are produced from this acetate pathway, and if it further get converted to butyrate, it will give only 2 moles of hydrogen. Because you can see here, the 2 hydrogen are used here for the further conversion of acetoacetyl coenzyme to butyryl. These 2 hydrogen are used here. So after the reaction, only 2 hydrogen molecule are obtained. So for this question answer is B. These type of question, like how many moles, here because you are studying about the hydrogen production or hydrogen fuel, here they are asking this, or these type of question, those who are from biotech background, they must know that in biochemistry they usually study these type of things, that how many molecules are produced from a particular component following this pathway, especially this is the glycolysis pathway. So you can study these type of things also. Okay, this is our next question. This step we have already discussed in the last session or last assignment. Okay, let's read the question. That the dark fermentation process involves the anaerobic breakdown of the high molecular weight organic substrate into. Okay, so this high molecular weight, here they have complex organic matter, either carbohydrate, protein, fat, whatever it is. During this anaerobic fermentation, or this you can also call this as dark fermentation, it usually occur in 4 steps: hydrolysis, acidogenesis, acetogenesis, and fourth is methanogenesis. Satvika, all of the above. Yeah, correct. So it finally forms all types of compound, all smaller molecules. Either you will get the volatile fatty acids, hydrogen, CO2, or methane also. So it will break down into volatile fatty acid, carbon dioxide, hydrogen. All these are correct. So for this question answer is D. Yeah. So this is next question. Which among the following can be called as a carbon neutral fuel? This is very simple question. So anyone please answer. Let's read option first.
Hydrogen.
Hydrogen, yeah. First one is methane, second is hydrogen, ethanol and butanol. So this hydrogen is a carbon neutral fuel, means it will not harm the environment without, like, the carbon balancing is there in the hydrogen fuel. When we use hydrogen fuel, it will not emit any gases or any harmful carbon compound. So, but there is problem with this hydrogen fuel. What is the issue with this? Normally, like at to present time, how we are producing this hydrogen gas, the production method use, the production method is not carbon neutral. Okay. So the production of hydrogen fuel is not carbon neutral, but when you use this hydrogen fuel, like suppose now you are using it for running any machine, so this will not produce any component. So this fuel is considered as a carbon neutral fuel, but its production process is not like that much advanced that it is become carbon neutral. So this process is under research, that many people are trying to do this. Even some people are producing hydrogen without using like making it as a carbon neutral process, but that is not at a very large scale. So in future this problem will be solved. So for this question answer is B, that hydrogen is a carbon neutral fuel. For this question answer is B. Okay, and this is your last question for today's assignment and this class also. You guys have asked me about the question in the last lecture material. At the last slide there is a name of two people are given for the TA, but I'm not sure that they are TA for this session also or not. So you might contact them or directly contact NPTEL for the same. Because the course is running previously, so I don't know whether those are the TA in this course also or not. So if anyone has any query, then please let me know. Or any other doubt.
Ma'am, I have some fundamental doubt.
Yeah,
How to distinguish between fungi and bacteria?
okay. If you, first one is like from the structure itself. Like if you like visual them, you can easily differentiate them. Like whatever. Which one is fungi and which one is bacteria? Because like if you ever if you have ever see them, like because suppose on a bread the fungus texture is very different, and when there is a bacterial infection, the structure or the topology become very different of these two. So first major difference is their structure itself. And their growth condition is almost similar for, like, normally that is similar, but for particular they are different, like different, but normally if it is a rodent thing, then both of these can grow there. So the very simple is either from the texture you will differentiate or from the previous studies, like
You mean both are single cell microorganism, no?
y- this fungi is a more complicated, actually, more type of cell, or you can say that is more advanced function this have as compared to bacteria.
Is there any way by name we can identify this is fungi or this is bacteria?
Okay,
Yeah.
from name, if you know few classes, like if you know few classes of bacteria, that Staphylococcus, like Mycobacterium species, if you know the species name, then you might differentiate. But if you randomly see any name, then you can't differentiate in through the name itself.
Okay.
Like if you know that it is from a Aspergillus family, then you will differentiate. But
The
if
Aspergillus family, that is surely fungi.
yeah, so like if you know the family name, then you can differentiate. But if it is given randomly, then you can't differentiate from the name.
Okay, ma'am. Thank you.
Okay. Okay, any other doubt? And fungi can be single cell, but it is also complex. But bacteria are single cell. So this is also.
Okay. Now, will you upload this in the problem solving section?
Yes,
In the
yes, I will upload it.
upload it as early as possible.
Yeah, yeah, I will upload it. Satvika is asking something. Ma'am, I have asked doubt in quiz 10 week. If an exam is secure, but assignment score is lower than classification. Okay, no, actually in the exam marks, they calculate both. The assignment marks are also considered and exam marks also. So you must submit the assignment on time, Ruchi. I have asked out in the. Yeah, Satvika, I remember that you said that not is given. So from that the question is marked incorrect. I have informed that about the question. Ruchi, you can even see this thing in NPTEL site that they have mentioned that how they consider the assignment and the final exam. So you have to submit. Can we get marks for that?
They will take best of 8 assignments and then average
Okay.
it, and then 25% of assignment
Okay.
marks are taken for final exam.
Hmm. Can we get marks for that? Okay, this thing I don't know, like because they will finally take the best score. So whether they will mark those question or not, that I can't say. Because the marking and all the things they did it by themselves. Okay, any other doubt? Okay, thank you everyone, and all the best for your exam. Do we have class next week? Okay, if there is a like revision class, then probably, and if it is not arranged, then it this is the last class. If the like NPTEL arrange the revision session for you, then there will be one class, and if the revision session is not there, so like till today I don't have any information about the revision session. For exam it will be MCQs. This I don't know, that exam it is MCQ or like mix, both type of things are there. Satvika, I don't know about the exam pattern, so like I can't help in this, like whether it is MCQ or not, or how many questions are there. This thing you can ask from someone who has given this exam previously, or it might change every time. So just try to read all the things, at least go through the every lecture material, because when once you go through all the lecture material, you will definitely be able to answer the things. Or if it's MCQ, then you can easily be able to at least uh, remove, not exact, if you don't know exactly the answer, then through the Gleason's method you can exactly get the correct answer. By removing the wrong answer, you will definitely get the right answer, because all the questions you have seen in all the assignments, like this is your 12th assignment, so in all the assignments questions are really simple. You just know a little bit of idea about the topic, and like clear about the some things, that what is particular thing is given, like some things you have to learn, but other things if you read it once only, then it will be fine.
But numericals is going to be challenge.
Yeah,
Like how much percentage of numerical questions will be there, and
definitely numerical will be there, but if it is, hopefully it will not be more, because in on the initial classes it has, we have numericals, but in the last assignments we don't have usually the numerical. In this in this last lecture you might have one or two numerical, but that is not very important kind of thing. Like I'm not saying don't study that, study that also, but the initially that where we have discussed about the stoichiometry, that part is really important for the numerical part. Okay, so everyone, all the best for your exam. and happy Diwali.
Thank you, ma'am.
Thank you.
Thank you. You are really very thorough in your subject, and the way you explain to us is fantastic in all through the all lectures.
Thank you
Thank
so
you very
much.
much.
I only tried this time, like this is my first time, so I'll try to next time it will be better, hopefully.